and welcome everyone to this first podcast on gene regulation. Over the next few podcasts, we're going to talk about some basic information on gene regulation. Then we're going to spend quite a bit of time talking about bacterial gene regulation. And then we're going to focus in on a particular operon called the lactose operon, or we'll just call it the lac operon. And then we'll close talking about eukaryotic gene regulation. As we talk about eukaryotic gene regulation, we'll also spend some time talking about epigenetics. But let's go ahead and get started. And in this podcast, we're going to talk about some of this basic information, and we're going to begin talking about bacterial gene regulation. On the next podcast, we'll talk about the lactose operon, and then the next two podcasts, we'll talk about eukaryotic gene regulation with epigenetics. Okay, let's go ahead and get started. Okay, let's talk about some of that basic information as it relates to gene regulation. If you remember in the last several podcasts, we've talked about something that we call the central dogma. And remember, the central dogma takes DNA, and through the process of transcription, makes RNA. And we've been focusing mainly on messenger RNA. But all RNA molecules are made through transcription. And then we also talked about modifying RNA, and mainly in eukaryotes. Now we have this modified RNA. And remember, we said before that has that poly A tail with that G cap. And again, remember, this is only seen in eukaryotes. Now, this modified RNA is next translated. And again, remember, if we're talking about bacteria, it would just go straight from this RNA to the translation step. And as that RNA is being translated, it makes a protein. Now, we didn't talk about this, but sometimes this protein by itself is inactive. And so sometimes there are steps that we'll just call post-translational modification, where that protein is activated. And so now it's an active protein. So various chemicals could be added to it, like a phosphate group, or an acetyl group, or a methyl group. And we'll talk about these later. Now, since the process of taking a gene that's on the DNA and making it all the way to this fully folded activated protein is a multi-step process, it's important to remember that gene regulation can occur at any one of these steps, as well as some steps that we didn't talk about. But this is a big picture of how that gene can be regulated, from the DNA all the way to the active protein. Now we're going to focus primarily on regulation at the transcription level, both in bacteria and eukaryotes. Bacteria primarily use transcription, whereas eukaryotes will use a variety of methods. But we're going to focus on transcription. And since we're going to focus on transcription, I want to talk about several proteins that can bind this DNA molecule to regulate it. And I shouldn't say I'm going to talk about several proteins. I'm going to talk about several parts of proteins. So if you see this protein down here, it's all folded up. But certain areas of it we call motifs. So maybe this part here is a motif. And a motif just means that's how that area of the protein folded. And based upon how it folds, it's going to interact with different targets. Now, these motifs come in a wide array of classifications. And three that I want you to know are helix, turn helix, zinc fingers, and leucine zippers. So you may have this large protein here, but part of that protein is going to fold in a way that equals one of these motifs. Each one of these motifs will bind to the major groove in DNA. And as it binds to those major grooves, it brings, of course, the whole protein into that area, as well as other proteins that might be attached to it. Many of these other proteins are going to help regulate the expression of this gene. The only thing else I want you to know on these is that bacteria primarily use these helix-turn-helix motifs. and these are seen in eukaryotes. There are similar helix-turn helixes in eukaryotes as well, but I would like you to know that bacteria primarily use these helix-turn helixes in their proteins to bind to the DNA. So these are the only 3 DNA binding motifs I want you to know. Now, proteins that have these DNA binding motifs, when they bind DNA, how are they going to regulate its expression? So let's move forward now and talk more specifically about bacterial gene regulation. Alright, so bacterial gene regulation. Now, as we begin, I want to talk specifically how bacteria organize their genes. Now, bacterial genes will regulate their genes by 2 special sequences. The first one, I'm going to put an O there, but it stands for operator. They also have a sequence that you've heard about before called a promoter, and I'll just put a P there. I should also remind you that what we're looking at here is just the way that we're describing the DNA, where the gene is located. But of course, we still have our 5', 3' ends, 3', 5'. Now, bacteria do something pretty convenient. They will cluster several genes together, and we'll just call these A, B, and C. And we'll call these open reading frames, or ORFs. Each one of these will go on to make a protein A, a protein B, and a protein C. What's different compared to eukaryotes is that they will regulate all 3 of these genes, and sometimes it's more than 3, but in this case I drew 3. They'll regulate all of these genes with a single promoter. As we will see when we move on to eukaryotes, it'll be 1 promoter, 1 gene. So what happens is, our friend from the past here, RNA polymerase, binds to the promoter, and it will transcribe these genes. And it doesn't transcribe them individually, it makes one long RNA molecule. And on this RNA molecule will be the open reading frames for genes A, B, and C. This messenger RNA then is translated. So let's go ahead and write transcription here, just so we keep everything on board. So then it's translated, and abbreviating it with a TLN. And as it's translated, now it will make 3 separate proteins. Protein A, protein B, and protein C. Now, bacteria doesn't just combine any 3 genes. They combine 3 genes that work in a similar pathway. So for instance, if these genes were important in making this substrate into this product, these proteins that were just made from this operon would all be involved in this pathway. So for instance, this protein A here might be what's needed to convert this substrate into this intermediate. And then protein B here might be important in making this substrate into this next substrate. And then protein C here is going to be responsible for making our final product here that I've already drawn. So in this way, what bacteria can do, and it's very convenient for them, they can turn on all these genes by having just one regulatory component. You activate here, it turns these all on in order to make a certain product. As we'll see, sometimes this substrate or this product is important in regulating this operon. Now it's important to make sure that whether or not this operon is turned on or off is highly regulated. There's no reason to make all these proteins if you don't need them. So what's going to regulate whether or not RNA polymerase transcribes these genes? It turns out that there are 2 other genes that are going to be important in regulating this operon, deciding whether or not to turn it on or off. These other genes are not part of the same operon, they're separate from it, and each of these promoters regulates a separate open reading frame. This one I'm going to call an R. And that's because the protein that it makes eventually, it'll make the messenger RNA, then the protein, but it's going to make a protein that we're going to call a repressor. This one down here is going to make a protein that we'll call an activator. Both repressors and activators are important in regulating whether or not this operon is turned on or off. Now I'd like to talk about how we classify the operons. I went ahead and erased it because I want to have some space here to describe how we classify operons. Operons can either be inducible operons or they can be repressible An inducible operon, usually inactive, meaning it is turned off, but can become active or turned on. Now, a repressible operon is usually active, meaning the genes are turned on, but it can become inactive, meaning the genes can be turned off. So to look at this, I want to look at an operon that is fully on. So on this operon, we have our promoter, and we have our operator, and then we have our 3 genes. And again, remember, it can be more than 3. I'm just showing 3. Now for this to be fully on, our activator protein that we talked about on the last slide has to be bound to the promoter. When the activator protein is bound to the promoter, it will recruit RNA polymerase. In addition, the repressor protein is not on the operator. In this case, it is fully on. Now next, I want to show an operon down here that is fully off. We'll also draw our promoter and our operator with our 3 genes. Now for it to be fully off, not transcribing these genes, the activator protein is not going to be attached to the promoter, but the repressor will be attached to the operator. Now let's go back to these 2 terms up here. An inducible operon is usually inactive, meaning it's this one down here, fully off, but it can become active. So an inducible operon would go from this state here to this state on top here. Usually off, but can be turned on. Now a repressible operon is usually active, so it's going to represent what we see up here on top. So it's usually fully on, but it can become inactive. It can be turned off. And so this is our repressible. Alright, now what I want to do is spend some time talking about inducible operons, how it is that we go from a fully off state to a fully on state, and then I want to talk about repressible operons, where we go from a fully on state to a fully off state. So I'm going to erase this and redraw parts of it here. Okay, so let's talk about inducible operons. And like on the last slide, I'm just going to draw the gene as a single line. Inducible operons, remember, they're going to usually be turned off. So we'll have a situation here where the promoter doesn't have the activator bound to it, and the operator will have the repressor bound to it. And we can draw the activator over here, not on the promoter. So how do we induce this operon to turn on transcription of these open reading frames that we see in this generic operon? We do it with a tiny molecule that we'll call an inducer. Inducers always work on inducible operons. Repressible operons use a different kind of substance. Now typically, in an inducible operon, we want to remove the repressor and add the activator. And so we need an inducer to do that. And typically there will be different inducers. So I'm going to call this inducer A, because it's the one that's going to bind to the activator, or inducer R, because it's going to be the one that binds to the repressor. Now what happens is when inducer A is added to the system, it binds to the activator. When that happens, something called a allosteric, or sometimes we'll call it a conformational change occurs. And that conformational change now allows this activator to bind to the promoter, but only because the inducer A here has bound to it. This other inducer, remember, this other inducer is going to be a separate chemical. It's going to bind to this repressor. Again, the same kind of an allosteric conformational change occurs, but in this case, it causes the repressor to fall off. And so let's go ahead and erase the repressor from the operator here, and we'll erase this over here too, because these inducers have come into the system, allowed the activator to bind to the promoter, pulled off the repressor from the operator. And now what happens is RNA polymerase can now bind to this promoter and transcribe A, B, and C. When this repressor was originally bound here, it serves kind of as a roadblock. The RNA polymerase can't go past a repressor when it's bound to the operator. So as long as the repressor's there, RNA polymerase cannot go any further. So to turn a gene fully on, an operon fully on, you have to remove the repressor and you have to bind the activator. The activator recruits the RNA polymerase, and now it can transcribe these genes without being blocked by the repressor. Now let's talk about a different operon, an operon that's going to be a repressible operon. I want to be clear that operons are either inducible or repressible. They're not both. They're either one or the other. So this is an inducible operon. So let's draw a repressible operon next. All right, so a repressible operon. So here's our operon. We're going to have our promoter, like we always do. We're going to have our operator, and we're going to have our open reading frames here. And just to kind of drive home the point that these are different operons compared to the inducible operons, we'll just, instead of calling them ABC, we'll call them X, Y, Z genes. It's a repressible operon, so repressible operons are usually on, but can be turned off. But let's draw its usual state. So its usual state is that the activator is going to be bound to the promoter, and because of that, it's recruited RNA polymerase to transcribe these genes. And it can transcribe these genes, remember, because the repressor is not bound to the operator. So these genes are going to be turned on, making the proteins X, Y, and Z. In order to turn this off, we need to pull off the activator protein from the promoter and bind the repressor to the operator. So just like an inducer was important for an inducible operon, a co-repressor is going to be important for regulating the repressible operon. And we're just going to call this CR for co-repressor. So a co-repressor could come along here and it'll bind the activator. When it binds the activator, it's going to pull the activator off the promoter. And when it does that, it's no longer be on the promoter, of course. And because of that, the RNA polymerase will not be able to bind to this area efficiently. So it too will fall off. Now RNA polymerases with some low efficiency can still bind here and turn these genes on, very low rate. But to make sure that doesn't happen, the repressor is now going to be bound. And the way the repressor binds here is it will bind to a separate co-repressor. And when it binds that co-repressor, the repressor with the co-repressor added to it will bind to this operator. And when it binds to the operator, we've effectively turned this operon off. We're preventing the activator from binding to the promoter, preventing RNA polymerase from binding, and then the repressor binds here also to make sure any RNA polymerases that happen to bind here by chance don't turn these genes on. You need both to happen in order to efficiently turn off this kind of an operon. Just like with inducible operon, you needed both the activator to bind to the promoter and the repressor to fall off. Okay, that ends this podcast. In this podcast, remember, we talked about some general terms associated with regulating genes. We talked about the different levels of gene regulation, and to do that, we talked about the different stages of the central dogma, realizing that at each point of protein production or gene expression, a gene can be regulated. We then spent most of the podcast talking about bacterial gene regulation. In that, we talked about the nature of the operon, and we talked about specifically inducible versus repressible operons, and we talked about the role that repressors and activators have in both inducible and repressible operons. And we talked about how chemicals known as inducers function in inducible operons and how chemicals called co-repressors function in repressible operons. In the next podcast, we're going to talk in much greater detail about a specific bacterial operon called the lac operon. If you have any questions over this material, please make sure you come see me. If not, I'll see you in class. Bye.